Hi, I'd like to thank the organizers for selecting my proceedings talk, Boosting the Accuracy of Protein Secondary Structure Prediction Through Nearest Neighbor Search and Method Hybridization, to appear in this session. This work was done by Spencer Krieger and John Kasesioglu. I'm Spencer Krieger, and I'm excited to share this work with you today. The task we address in this talk is protein secondary structure prediction. Protein secondary structure is a local and discrete form of protein structure. Given an amino acid sequence, each amino acid is assigned one of 3 or 8 secondary structure classes, depending on the model. The task is, given amino acid sequence P, predict the corresponding secondary structure sequence S of the folded protein. For each amino acid position in P, S gives the corresponding secondary structure state where E stands for beta strand, C stands for coil, and H stands for helix, where this example is using the three-state model. This is a classic problem in bioinformatics that has been well studied, with over 45 tools being produced over 60 years. Of currently available tools, state-of-the-art methods typically perform SciBlast homology search over large protein sequence databases to find homologous sequences. These databases lack protein structure, even though there is a large number of proteins with known structure. SciBlast over the largest sequence database used by a state-of-the-art tool can take as long as 30 minutes. Many of these tools do not output physically valid secondary structure. In particular, they don't satisfy minimum run lengths. For example, an alpha helix must be at least 5 residues long in nature. These methods report confidences, which are a measure of how unambiguous the predicted structure class is at a residue, which doesn't translate to estimated accuracy, which is more desirable to a user. In contrast, our new tool NESI is an algorithmic approach to protein secondary structure prediction, which avoids expensive SciBlast database homology searches. NESI is a template-based tool and uses k-nearest neighbor search over a smaller template database of fixed-length protein words with known secondary structure. These words are searched under a distance metric that obeys the triangle inequality, so we can leverage algorithms for nearest neighbor search over a metric space, so the problem is much more structured. NESI also computes a maximum likelihood physically valid prediction via dynamic programming, where the dynamic programming satisfies minimum run lengths, even for the 8-state model, and also runs in linear time and is easily adapted to other tools. Instead of outputting confidences, NESI uses a feature-based accuracy estimator that takes a feature of the prediction process and estimates the unknown true accuracy of its prediction. We combine our template-based tool NESI with a template-free tool to yield a hybrid method that gains the advantages of both tools. This hybrid method exceeds the state-of-the-art accuracy by more than 3% for the 3-state model of prediction and more than 7% for 8-state prediction. Next, we'll give an overview of NESI's prediction process. Given an input amino acid sequence in our preprocessed template database, we perform nearest neighbor search on words from the input sequence to find their nearest neighbors from the template database. These nearest neighbor words are given to an overlapping words procedure that combines the secondary structure sequences from the template words to estimate structure state probabilities at residues. These state probabilities are fed into a dynamic programming algorithm that does not output a greedy prediction, which would simply choose the state with highest probability, but instead computes a physically valid structure of maximum likelihood. This maximum likelihood prediction is then output by NESI. To get residue state probabilities, we use the following procedure. Given an input protein sequence, we extract fixed-length words at every position, where each colored box represents a different fixed-length word. To obtain state probabilities for this residue highlighted in red, for each word that contains this residue, nearest neighbors are found from our template database using the dispersion tree data structure. These template words contribute to the state probabilities of each residue they cover, where the structure states are indicated by the colors of the letters, where alpha helix is shown in blue, beta strand is shown in green, and coil is shown in black. So for the highlighted residue, the frequencies of structure states of the template words at the boxed positions determine its state probabilities. And for this example, the frequencies of alpha, beta, and coil give the structure probabilities 0, 0.62, and 0.38. These state probabilities are then used in a maximum likelihood prediction procedure. The procedure considers physically valid secondary structure sequences that satisfy minimum run length requirements of structure classes. We find the optimal physically valid structure sequence of maximum likelihood by dynamic programming. The maximum likelihood objective function that this algorithm optimizes is the following. The function has the form of a product, where the first factors in the product come from the structure probabilities previously calculated. For a protein sequence of length n, there are n factors, one for each residue position in the protein. This factor is the probability that the predicted state in S occurs at that residue. This next set of factors deal with transition probabilities, or the probability that one structure state is followed by another structure state. And the last group of factors deal with run lengths and use the empirical distribution of run lengths observed in real proteins. We minimize the log likelihood of this objective function by solving a recurrence equation. The dynamic programming algorithm computes the quantity L of ILC, which is the negative log likelihood of an optimal prediction ending at position I with structure class C and run length L. For example, for the general case of L greater than the minimum run length of class C, LILC satisfies the following recurrence equation. L of ILC is equal to a coefficient multiplied by the probability that structure class C occurs at position I, plus a transition coefficient multiplied by the probability that structure class C transitions to C at position I, or here, where no transition takes place, plus a run length coefficient multiplied by the probability that the run of structure class C is extended by 1 at position I, plus the quantity L of I-1, L-1, C, or the negative log likelihood of an optimal prediction ending at the previous position, a run length 1 smaller, and with the same structure class C. This shows just one of 3 cases of the full recurrence equation for the dynamic programming algorithm. The complete dynamic programming algorithm is fast. Given a sequence of length n, it runs in linear time. A unique aspect of our approach is that it uses an accuracy estimator, which estimates the unknown true accuracy of its prediction based on an easily computable feature. The feature we use is average word distance of the nearest neighbors from the template database, found for a residue by the overlapping words procedure. In this figure, the horizontal axis is average word distance at a residue, and the vertical axis is the average accuracy of residues at that word distance. Each circle represents a residue from CASP proteins, which is plotted at its average word distance on the horizontal axis. Its value on the vertical axis is average empirical accuracy of residues with similar word distance. The dashed lines give the piecewise linear fitted estimator, which maps word distance to estimated residue accuracy. To get the estimated accuracy of the prediction for the entire protein, we average this estimate over its residues. The resulting accuracy estimator has an average error of 5% for the 3-state model and 8% for the 8-state model. To give an illustration of the behavior of this estimator, this figure shows a plot of true accuracy versus estimated accuracy. Each circle here represents a protein from a benchmark dataset where the estimated accuracy of Nessie's prediction is given on the horizontal axis and its true accuracy is given on the vertical axis. This dotted line is the identity function, where estimated accuracy equals true accuracy. Note that above 80% estimated accuracy, there is a tight cluster of proteins of high true accuracy. This leads to a natural threshold for a hybrid approach to prediction, which is explained on the next slide. This hybrid approach combines NESI, a template-based method, with a template-free method. Given NESI's template-based prediction and the prediction of a template-free method, we estimate the accuracy of NESI's template-based prediction. Using the threshold shown previously, if the estimated accuracy is above the threshold, the hybrid outputs NESI's prediction. Otherwise, it outputs the alternate prediction of a template-free tool. The hybrid gains the advantages of a template-based method, where when a good template is found, the prediction has high accuracy. We combine NESI with a robust and stable template-free method, so when no good template is found, the accuracy of the hybrid prediction is robust and stable. Now we'll go into experimental results. We evaluated NESI and other tools on CASP and PDB benchmark datasets, where the PDB datasets take a random subset of proteins from PDB for each year between 2015 and 2019. For each evaluation, NESI uses a template database formed only from proteins available before the date of the CASP competition or from before the year of the PDB dataset. NESI's parameters were tuned using a testing dataset that contains none of the evaluation proteins. We compare against the following state-of-the-art tools. JPRED and CyPRED are some of the earliest tools still in widespread use. CyPRED pioneered using a CyBLAST homology search to form a position-specific scoring matrix for the input amino acid sequence, combined with a neural network to form the output prediction. SSPro is one of the only template-based approaches that searches for matches in a template database, and if none are found, relies on a CyBLAST search and neural network similar to CyPRED and JPRED. Porter and DeepCNF use homology search and variations on deep convolutional neural networks. Lastly, PSRSM uses semi-random subspaces, which is a variation on support vector machines. We now give prediction accuracies for these tools, including NESI and the hybrid method. The following table gives accuracies for the standard three-state model of secondary structure prediction. We use Q3 accuracy, which is the fraction of residues with correctly predicted secondary structure. The full proceedings paper gives accuracies on each CASP and PDB dataset, where here we show just the average over all datasets. We show the accuracies of these tools in increasing order of average accuracy, along with the hybrid boost, which is the increase in accuracy when a given tool is hybridized with NESI. PSRSM is the tool with highest average accuracy at 85.6%. Hybridizing PSRSM with NESI, which is the best hybrid, yields an increase in accuracy of 2.6% over the accuracy of PSRSM. Here, we show the accuracies for the 8-state model using Q8 accuracy. We give Q8 accuracies averaged over the CASP and PDB datasets for each tool and also their hybrid boost. There are only 4 tools that do 8-state prediction. We show them in order of increasing average accuracy. NESI has the highest average 8-state accuracy at 73.3%. Hybridizing each tool with NESI yields a significant boost in accuracy, where the best NESI hybrid is with Porter, and it achieves 80.4% accuracy, which is more than 7% higher than the best single tool. We now show a visualization of the predictions chosen by the best hybrid, which combines NESI and Porter. The horizontal axis corresponds to CASP proteins ordered by the accuracy of their hybrid prediction. The vertical axis is the Q8 accuracy of a prediction. At each protein rank on the horizontal axis, there is a blue circle and a green triangle corresponding to the Q8 accuracy of the NESI or Porter prediction. The black curve gives the accuracy of the hybrid method, which passes through the prediction it chooses for each protein. Any marker above the curve is a protein for which the hybrid did not make the optimal choice. Note that there are very few markers above the curve. The average accuracy of Nesse, Porter alone, and their hybrid are shown by the dashed and dotted lines. Note how much the hybrid exceeds the accuracy of both tools. We can bound the accuracy of an optimal hybrid method by considering an oracle that always outputs the prediction with higher true accuracy of the two hybridized methods. The following table gives the average accuracy of the best hybrid and its corresponding oracle. For the 8-state model, the best hybrid uses Nesse and Porter. It achieves a hybrid accuracy of 77.7%, which is only 0.4% below the oracle accuracy. The fact that it is much higher than a random selection shows the strength of the accuracy estimator. For the 3-state model, the best hybrid uses Nesse and PSRSM. Once again, the hybrid is only 0.6% below the oracle and far exceeds random selection. The hybrid approach almost matches the oracle accuracy on all datasets and even meets the oracle on some. In conclusion, our core approach to secondary structure prediction is an algorithmic approach that uses nearest neighbor search over a metric space, and in contrast to other methods, outputs maximum likelihood physically valid predictions. The hybrid approach exceeds the state-of-the-art accuracy by 3% for 3-state prediction and 7% for 8-state prediction. This opens up several lines of investigation for future work, such as constructing accuracy estimators for other tools besides Nesse, and generalizing the hybrid approach to accommodate multiple methods beyond the current version, which hybridizes just 2 methods. Our new tool Nesse, which implements our core approach, is available at nesse.cs.arizona.edu. We would like to thank the following people and the National Science Foundation for our funding. A poster on this work will be presented on July 15th and 16th from 7:45 a.m. and 9:15 a.m. Eastern Daylight Time. And I would like to thank all of you for listening to my presentation.